we're going to talk a lot about just, so let me just say, recap a little bit where we are. We talked about, a lot of the basic types, data types that we use in bioinformatics. Talked about some simple pipeline operations, variant calling, processing functional genomics data. We talked about making a data matrix, and the sort of big group of techniques that we do on the matrix, what supervised techniques like SVM, decision tree, unsupervised stuff. We talked a lot about hierarchical clustering. We talked about LDA, we talked about SVD, all the things we can do in an unsupervised fashion to explore our data matrix. Now what I'm going to do is, in a sense, zoom in on one type of analysis with the data matrix in detail. That's where we make it into a network. Okay, so we kind of make a kind of correlational plot, and then we threshold that, and we make a network. And there's a whole set of analyses that people do on networks that I think are quite useful. And so we're going to talk about networks now for a little bit. And hopefully we'll get through most of this stuff. Okay. So first, I just want to motivate networks. And so, I think networks, I think kind of provide a really nice halfway house for biological data or lots of data. And how do we think about that? Well, in genomics, often we have, of course, the notion of genomic coordinates, which we know is like one part of our matrix, and here's the other part, the different assays. But, it's in a sense, it's not a very rich representation in terms of how the bits of the genome relate to each other. They're just splayed out here on a line. And, if we imagine having tons and tons of assays, which is what these genomic data matrices look like, we really have to wonder, how does this type of view of things give you an overall view with thousands of tracks? These different assays are usually referred to as tracks. So a lot of people say that this view is a kind of very linear view, a kind of partsless view. We have all the different pieces, these are different genes, but we don't really know how they work together. Now, kind of ideally where we want to go in biological science is deep mechanism. Now, deep mechanism is not really encapsulated by these genomic views. What it really says is each of these transcription factors and sites on the genome and so forth, of course, is a three-dimensional molecular structure that we're going to be talking about later in the class, and really thinking about how these structures work together as physical objects and so forth. Now, this is very complicated. We do not have a view of all the molecules in the human genome to this level. This picture, for instance, shows the conformational change in the ribosome. This is elusive. Now, one day, maybe in 20 years, 50 years, we will have a detailed mechanistic view of every molecule in the cell and really understand how the cell works as a machine. That's not where we are now. On the other hand, this kind of track view or partsless view is very not rich. And so I think the network view, its power is it kind of sits midway between here. It provides some kind of connections between all these different things together, but it doesn't force this level of structure on things. And because of that, it's very useful. And people use the term wiring diagram, pathway diagram. These diagrams, I think, are very evocative and very useful for people for giving them intuition. Now, I think also, a lot of times people think, when they think about diseases, a lot of times say the gene associated with this, the gene that a drug acts on. That's true to some degree, but if you really think about a lot of diseases like breast cancer or Alzheimer's or whatever, often it's really more about pathways, groups of genes, subnetworks that are dysregulated or doing things differently. And likewise, when we think about interfering with these things, it's often, again, a small molecule that's maybe acting on a number of things within a pathway or affecting a whole pathway, or maybe a molecule that's disrupting an interaction or really literally acting on an edge that's connecting two molecules. So again, sometimes this type of view is quite useful. And here's a picture. There's a lot of reviews on this interactome methods in human disease. This is some papers that take that view. Now, I think another satisfying aspect of networks is that they're used in many different communities, and there's almost a network science language and almost a network science world that spans different communities, and that provides a powerful unifying view on science. So for instance, of course, we talk about protein interaction networks and molecular networks here, but of course, within biology, there's also neural networks. So these are networks in the wiring in our brain. There's food webs. There's networks we now hear a lot about in terms of epidemiology, in terms of disease spread when we model, say, the movement of coronavirus. More generally, we have social networks. We have a lot of intuition for these things. We have, of course, electrical networks and so forth. And, people in network science tend to study structure of networks kind of cross-discipline and look at different patterns. And one of the, I think, famous people in this discipline is this fellow named Barabasi, who's in Boston. And here's a book that he's published, Linked. It talks about, the new science of networks. And this was actually a new science 15 years ago. Now it's the old science of networks, but still there's quite a community of these people that think about things. And I think one of the most powerful aspects of this network science is that some of the subdomains that we can study networks in, we can get very good intuition about what's going on because they're very close to what we deal with. And we can transfer that intuition to another domain like the molecular biological domain that we have very bad intuition for. So let's look at this picture here for a second. Now that's actually an old picture. And I took these pictures from the New York Times maybe 15 years ago, but they're still completely representative. What was going on here at that time was, I think George W. Bush was the president at that time. And there was some scandal or something, I don't remember what it was. And the New York Times did a thing, and what they did, which was kind of cool, is they made arrows, and they had all these famous people. Here's Bill Clinton, here's some mayor or something like that. And they show how people are blaming other people. Okay. And, and then you can see George Bush is blaming himself. That's nice. But I guess the key thing is if you look at these arrows, you kind of get the notion of looking at where all the blame is going towards identifying the causal factor, the causal regulator in this disaster, whatever happened here. Now, you might say, well, that's obvious, but think about it. If we were just looking at a molecular diagram and these people's faces were not faces of people, but they were pictures of molecules, what this blame game, what these arrows would show us is where is the causal regulator for a process. And that's what we often study in network science, where we just look at these connections here and we say, oh, these things up here, they're somehow more regulating or more the cause of that process. And here's another famous one here. So I think, I'm not remembering exactly what this guy did wrong. These are crimes and so forth. So this guy is a governor and he did something wrong. I think this had to do with Barack Obama when he became president. And then these pictures show all the people that he worked with, okay? And it's again colored by, these are the people that were found guilty of certain things. So this guy, Rezko, was guilty of something. This guy, Levine, was guilty of something. And, the implication of this, if you look at this figure, was there's a group of people here that they're all kind of wrong. And so maybe the other nodes that are linked, like this one, maybe even the governor, that are linked by many of these nodes are also red, and they should also be guilty. And again, it's obvious, this is guilt by association. If many of your friends are guilty of something, maybe you're also guilty too, maybe not, but that's a normal social suspicion. Again, we have easy intuition for this in a social context. In a biological context, of course, this is a way we would assign function to things. So we might we might make a network of genes color them a certain color and say, oh, this gene here or that gene here maybe is somehow related to this red color because it's connected to it. Completely obvious. And again, I think our intuition from a social context helps us here. So that's, I think, one really powerful aspect of how we can transfer intuition networks. Another aspect, which is more biological, is how we can represent many different biological processes on the same diagram with, say, arrows of different colors. And so this just picture shows lots of different genes, and they're connected by metabolic pathways, transcriptional regulatory network, protein-protein interactions. And the key idea is this type of diagram starts to hint at the biological complexity of what's going on without that too much detail that we get in the true mechanistic description. So a very useful representation. And then I guess the other thing the network science community really talks a lot about is that there's a whole world of people that draw networks and represent things visually very well with networks. And here's some nice ones. This is a website that has lots of nice visuals of these things. So a little bit about networks that we work on, just as introduction. We're going to mostly focus on the molecular networks that we have in biological science. What are they? Protein-protein interactions. A subgroup is phosphorylation, metabolic networks, pathways, and so forth. Regulatory networks, we talked about earlier how they come from ChIP-seq experiments, how we can build up networks. And I think one of the key things to realize about networks is the squared size of these networks. So for instance, in yeast, which has about 6,000 genes, we can make 18 million, 6,000 choose 2, combinations of these things. And these networks get bigger, as we get more parts, they get bigger in a squared sense. So they get potentially more complicated. There's a picture again of all our different types of networks. And one of the key aspects of these networks is some of them are what's called undirected networks, where we just have things connected, like for instance, the protein-protein interaction network. Other networks are what's called directed networks. For instance, in the transcriptional regulatory network, we have the notion of one thing regulating something else. Okay,